All right, for some reason the Zoom meeting ended and I had to log into Zoom again. We'll have two lecture meetings today. Not sure what's going on. Let me just quickly look and see if I can determine what's going on here. So I'm not sure. This is not supposed to happen. I'm looking to see what's going on here, why the meeting ended. I'm not going to change it now, but I'm just looking to see. Hmm, It's not even showing me. I don't know if you're seeing my screen. It's not even showing me the times of the meeting when I go in to edit the meeting. I'll have to look at that. Now, why is there a passcode? That's not my code. I don't understand this. All right. For some reason, the Zoom meeting is not working correctly, and I'm not sure what's going on. Fortunately, the lab meeting earlier today worked well. I'll look into that between the.
Actually, just got an email from Clark saying that there is an issue with Zoom meetings and classes timed out after 40 minutes, and our IT service team is working on it.
Oh, thank you. I didn't check my email, so that explains it. It's nothing I've done. It's something that's going wrong with the either Clark's connection to Zoom or Zoom itself. I don't know, but it's not. It shouldn't be something that I did. Anyways, we're back in a meeting. You're going to have two lecture Zoom meetings today, and my apologies. I don't know what's going on, but there is a problem with the Zoom, and I noticed that it gave me the 10-minute warning, and like I said, I went in there to change the meeting for the times, and I can't even find the time in the meeting. So I'll look into that in between the lecture and the lab. Anyways, let's go back to where we were. Up, I need to share my screen, don't I? Let's go there. My computer is going to be slow because it's processing the first Zoom meeting. Oops, that's not right. Let's go there. All right. So I mentioned that the carbon atoms in an organic molecule make a carbon skeleton backbone, but most of the molecule is held together by the carbon-carbon backbone. There are functional groups on organic compounds where there's a more than one atom attached to the carbon, and one functional group is OH minus. Functional groups give this molecule different properties than, let's say, methane alone. For example, methane is a non-polar molecule. It doesn't like to dissolve in water. It, oh, that's good enough. Uh, if we then attach an OH group to the methane, we call it methanol because this is an alcohol. It has an alcoholic group on it. And this methanol can dissolve in water. It also is no longer a nonpolar molecule. It's now a polar molecule because the OH group is polar. Any question about any of that? These functional groups influence the molecule and give the organic compound different chemical properties from, from the functional group. Any question about any of that? Sorry, I sneezed there. So the functional groups are responsible for most of the chemical and physical properties of a particular organic compound. Hydroxyl group is an alcohol, changing the molecule from nonpolar and hydrophobic to a molecule which is polar and hydrophilic. And then that allows the molecule to dissolve in water. You might not be familiar with methanol, but I bet everyone's familiar with ethanol. That's the alcohol you can drink. All right, here are some functional groups. You need to be familiar with the groups in red boxes. So you need to be familiar with an alcohol. It's in lipids, carbohydrates like sugars, in proteins, in nucleic acid. It's actually in all of them, all of the biological molecules, all the four biological molecules. The next functional group I want you to know is an amino group. It has an NH2 group. You can find amino groups in amino acids and proteins. The next group I want you to be familiar with is the carboxyl group. It is a C with a double O OH group attached to it. So you can call that COOH. Realize that first O is a double bond between the C and the O. All right, the first lecture file is just finished, so my computer will now start working a little faster. And then the last functional group you need to know is the phosphate group, PO4, found in ATP, DNA, RNA. Why do I think I want you to know the functional groups found in the red boxes and not the other groups like this ketone group or the methyl group? Anyone have a suggestion? Why do I want you to know like the carboxyl group and the amino group and not the other groups like the methyl group, the ketone group? No one has a suggestion. It's because the groups shown in red are the groups important for giving properties to the four uh, molecules of life: proteins, carbohydrates, lipids, and nucleic acids. So these groups we'll run into time and time again when we're talking about the four biological molecules. And then the ketone, we won't run into that one very often. You should be familiar with the groups and that some of the groups can be on the same molecule. Like an amino acid has two functional groups attached to it. It has an amino group on one end of the amino acid and a carboxyl group on the other end of the amino acid. Any question about any of that? All right. You should know that organic molecules can combine into large molecules called macromolecules. The macromolecules are usually polymers consisting of many small repeating molecules called monomers. For example, glucose is a monomer, and then the polymer would be starch, and starch is just many glucose molecules hooked together one after the other to make the starch. So starch is both a macromolecule and a polymer made up of many repeating monomers that are, in this case, glucose. Any question about any of that? Three of the biological molecules are macromolecules made up of polymers, and that would be the carbohydrates, the nucleic acid, and the proteins. They are all macromolecules, meaning polymers made up of monomers. The fourth biological molecule, the lipid, some lipids are a macromolecule, meaning some are polymers made up of monomers. But other lipids are not polymers. Like a wax is a lipid, but it is not a polymer made up of smaller repeating monomers. Any question about any of that? And then an example of a lipid that is made up of monomers would be a triglyceride, which is simply a fat you can find in an animal. And you look at your belly, you'll see lots of triglycerides. Even if you're thin, you still have the triglycerides there. And uh, a fat or a triglyceride is made up of three fatty acids and one glycerol molecule. So those are the monomers of a triglyceride. Any question about any of that? All right. Organic compounds can be synthesized by taking a monomer and joining them together where this O binds to that R so that we have actually I take that back. That R binds to that R, that OH comes off and the H comes off forming water. And that we call a dehydration synthesis reaction where we make a larger molecule, synthesis, from two monomers to make, actually that would be a dimer. You get several of them together, you get a polymer. And then water is made. And that's the dehydration synthesis reaction, also called the condensation reaction. Any question about any of that? Hydrolysis is the reverse, where you take a polymer or a dimer, you add water, the water splits, the H forms, breaks off the water and forms onto this R, and then the OH group forms onto that R, and then that bond breaks, and then we have ROH and HR to make the monomers. And that is called hydrolysis. Any questions about any of that? You should be familiar with hydrolysis if you've seen the movie Back to the Future 2, when Marty McFly says to his mother, "Ma, you sure know how to hydrate a pizza," when she took this black hockey puck round thing and put it in her hydrolysis machine. She was undergoing hydrolysis, and then out of the machine came this beautiful looking pizza. Anyone remember that? That's an example of hydrolysis. Any question about any of that? So hydrolysis is just the reverse of dehydration synthesis. And remember, dehydration synthesis is also called the condensation reaction. I usually call it dehydration synthesis because you're synthesizing a larger molecule. And it's called hydrolysis because you're using water to split the larger molecule into the smaller molecules. Here's another example of dehydration synthesis where we have a monomer adding to a smaller polymer. The OH comes off the monomer, the H comes off the smaller polymer. The monomer here then binds to the polymer there, and then the water forms, and that's dehydration synthesis. An exact example of this would be when you eat a lot of sugar or glucose and it breaks down to glucose into your blood, and then the glucose is added to glycogen in your liver. The glucose will be added to the glycogen, and then you get a larger polymer, a larger glycogen molecule, because the monomer of glucose has been added to it, and then water is formed. Hydrolysis is when water splits a polymer into, in this case, a monomer and a smaller polymer. The H comes off the OH of the water, splits the monomer, the H joins the polymer, the smaller polymer, and then the OH of the water joins the monomer. and that is hydrolysis. How Marty McFly was able to eat the pizza when his mother hydrated the pizza. Any question about any of that? All right, let's talk about the four biological molecules. Carbohydrates include sugar, starch, and other complex carbohydrates. Carbohydrates are an important energy source. You can find them in the structure of your cell, like on the cell surface, on the structure of your body, can be carbohydrates making it. You can find carbohydrates in nucleic acids. Carbohydrates always consist of carbon, hydrogen, and oxygen, and not just any combination of carbon, hydrogen, and oxygen, but the general formula for each carbon, there's two hydrogens and one oxygen. So for each carbohydrate, you can check CH2O and then put a bracket around it and then an N. And in the case of glucose, you have that N equal to 6. So the glucose is C6H12O6 for the carbohydrate. So just remember the general formula of a carbohydrate is CH2O with an N to the whatever that's called, subscript N. And for like glucose, you put in a 6 for that N. Simple sugars, or strings of simple sugars, linked together are simply what a carbohydrate is. We can classify carbohydrates into three major groups based upon their size. If there's only one monomer in the carbohydrate, we call that a monosaccharide, which is also called a simple sugar. So glucose, fructose, beta-galactose are example of monosaccharides. You can have a carbohydrate composed of two monomers, and that's a disaccharide. An example of a disaccharide would be table sugar, also called sucrose, maltose, lactose, the sugar in milk. Those are all disaccharides. And then there's polysaccharides, and these consist of many monomers. Poly means many, saccharide. A saccharide literally means sugar. And we're talking about simple sugar here for when we use the term saccharide, because that's the monomer. And starch is an example of a polysaccharide. All starch is glucose hooked together one after the other. There are other starches, like glycogen found in your muscles. We'll talk about cellulose later in this lesson, another polysaccharide carbohydrate. Any question about any of that? All right. So here we're looking at monosaccharides, glucose and fructose. They can get together in a, dehydration synthesis reaction where the H comes off the fructose, the OH comes off the glucose, and then that carbon right there binds directly to that oxygen on fructose. So that carbon is binding to the oxygen to make a disaccharide, and this disaccharide is actually sucrose, table sugar. And then water comes out because of the water coming off the monosaccharides. And that is a dehydration synthesis. Monosaccharides are always simple sugars between 3 and 7 carbon atoms, but most monosaccharides are between 5 and 6 carbons. Like all simple sugars have 5 or 6 carbons. Glucose and fructose each have 6 carbons. We'll talk about ribose and deoxyribose later, I think in this lesson, but maybe in a future lesson. Those are simple sugars that only have 5 carbons. Most monosaccharides that we eat have 6 carbons, but there are some that have 5. Monosaccharides are sweet. They're all water soluble. A disaccharide is formed when you combine 2 monosaccharides, such as sucrose, and then you can split a disaccharide, such as sucrose, by adding water. The water breaks apart. OH minus group adds to the glucose, and then the H of the water adds to the fructose, and then the disaccharide by hydrolysis will break down into fructose and glucose. Any question about any of that? All right. Polysaccharide consists of many monosaccharides. The chain can be straight or branched. A starch is mostly, it's not entirely, but it's mostly a straight chain of glucose hooked together one after another. Examples of polysaccharides include starch, glycogen, dextran, cellulose. All are glucose polymers where glucose is chemically bonded covalently between one glucose molecule and another. Here are some starchy foods. Everybody knows the bread, cereals, pasta, rice, and potatoes are starchy food. But beans and chestnuts are also starchy food. You look at beans, about 1/3 of the weight of beans is due to carbohydrates, 1/3 of the weight to fat, and 1/3 the weight to protein. I don't know the exact chemical makeup of chestnuts. I would assume a chestnut has a little more carbohydrates and maybe more protein than it does lipid, but definitely more carbohydrate than lipid in the chestnut. Then beans, but beans about 1/3, 1/3, 1/3. But it is a starchy food because 1/3 of it is starch. Any question about any of that? All right, let's talk about some other um, polysaccharides. Go to the bottom here. There you see starch. Go to the bottom here. It's mostly glucose hooked together in a linear arrangement to make the starch. There are a few branched structures in starch. You don't need to know that. A glycogen is similar to starch, except that the glucose is held together one after the other, meaning the monomer is, but it's not a linear chain. The glycogen branches. Like right here, we have glucose, glucose, and then right there, the chain branches into two, one branch being glucose here, and the next being glucose there. And glycogen just branches, well, branches all the time, much more commonly than in starch. Like I said, very rarely do you have a branch in starch. Cellulose is also a carbohydrate, which is glucose hooked together one after the other. Let me blow this up as great as I can. Oop, I can't blow it up any further. Let's see if I can blow it up if I end it this way. There we go. Um, so the glucose is held together with the blue lines, and that's a chemical covalent bond holding the glucose together one after the other. In cellulose, we have several rows of glucose, all held together each row by a chemical covalent bond, meaning this glucose is held to that glucose and that glucose by a chemical covalent bond. But this glucose here has a hydrogen bond to that glucose there and a hydrogen bond to that glucose there. And as you can see in cellulose, we have a lot of hydrogen bonds linking the glucose together, meaning the glucose over here is linked to that glucose over there, as well as that glucose over there, meaning this glucose has two hydrogen bonds to the glucose. Any question about any of that? Because of all the hydrogen bonds in glucose, it is very difficult to digest cellulose. Humans cannot digest cellulose, so we cannot eat grass and break it down into glucose and be something we could use as a food source. This cow can, however, the cow doesn't actually break down the grass either, the cellulose in the grass. What the cow's stomach does is it has a microbe, it actually has two microbes. We'll just talk about one for the moment. And it is the microorganism that breaks down the cellulose, breaking it down into glucose. And then the microorganism shares some of that glucose with the cow. And that's how the cow can eat grass, because the grass will contain most of the cellulose, and then the cow can digest the cellulose, not directly, but because it has a microorganism in its stomach, or I should say actually four of its stomachs, because the cow has four stomachs, to break down the cellulose. And then the cow can get some of the glucose from the microbe that actually breaks down the cellulose. Any question about any of that? All right, I'm not going to talk about chitin. I'm not going to talk now about peptidoglycan. We'll talk about peptidoglycan later in the term. Um, Peptidoglycan is both a carbohydrate and it's also a peptide, meaning it does have peptides in it, short peptides. Peptides are the building block of a protein, and they're made by amino acids. A peptide is shorter than a protein, but my point is um, just a minute, um, losing the name of this one. The peptidoglycan is both a carbohydrate and peptides, and we're not going to talk about it now. I'll talk about it in a later lesson. Any question about carbohydrates? If not, let's talk about the second of the four major biological molecules, lipids. Lipids always consist of carbon, hydrogen, and oxygen. However, lipids are mostly carbon and hydrogen. They do have oxygen, but they don't have very much oxygen. Lipids are mostly nonpolar and insoluble in water, meaning lipids are hydrophobic. There is one exception to this, and we'll talk about it later in this lesson in a slide or two, maybe three. But most lipids are hydrophobic and insoluble in water, and that's because they're nonpolar. Lipids are structural molecule in our cells and in our body. They also store energy. There are various roles for lipids in our body. Lipids are the primary component of the cell membrane, meaning it is a lipid that's the primary molecule in your cell membranes. Any question about lipids so far? All right, we can break lipids into simple lipids, complex lipids, and steroids. A simple lipid is like fatty acid. It's a major component of many lipids. It consists of a long chain of carbohydrogen, of carbon and hydrogen. Let me blow that up here and then scroll down to it. Gosh, this meeting's going to end in 10 minutes. Well, that'll be fine. I only need 7 more minutes. Um, so there's the fatty acid, mostly containing carbon and hydrogen. Does have an oxygen right there. You have two types of, oh, come on here. Fatty acids. You can have a saturated fatty acid, and that's where all of the carbon-carbon linkings happen by one covalent bond between the carbon. And you can have an unsaturated fatty acid where there is at least one carbon-carbon bond, which is two links between each carbon, meaning there's two covalent bonds between each carbon. Let me show you in the next slide an example of that. Blow this up so you can see it. This fatty acid has a double carbon linkage, meaning two chemical covalent bonds link the two carbons together. Wherever that happens, there's a kink in the chain. Like wherever there's a single carbon bond, the fatty acid is straight, and wherever there's a double carbon bond, we have a kink in the chain. You only need one double carbon bond in the entire molecule to call that molecule unsaturated. So a fatty acid right here, that's an unsaturated fatty acid. That is a saturated fatty acid. But in this lipid, meaning the whole lipid molecule here, there's only one double carbon bond. So this molecule is an unsaturated fat or lipid. Any question about any of that? You should know that unsaturated fatty acids or lipids are better for your heart than a saturated fat or lipid. And that's all I'm going to talk about it now, for now at least. A simple lipid called a fat or a triglyceride is three fatty acids linked together to glycerol. And then the H comes off the glycerol, the OH group comes off the fatty acid. And you have three of these fatty acids linked to the glycerol to make a triglyceride. And a triglyceride, or any fat, is a polymer made up of monomers, in this case, three fatty acids and one glycerol molecule. And as I stated, this triglyceride is unsaturated fat because it has one double carbon bond linking two carbons together. Triglycerides are the body's richest energy source. It's stored as adipose in animals. You can just look down at your belly and you'll find some triglycerides. By the richest energy source, it means you can get the most energy per pound or per gram, whatever you want to say, out of, let's say, per pound, from any other food that you could eat. You'll get the most energy from a triglyceride or a fat. That might not be the healthiest thing to eat, but let's say you're in Point Barrow, Alaska in the wintertime, you're driving your car and your car breaks down, you have to walk 20 miles through the winter weather to get to Point Barrow, and you want to carry some food with you to eat to give you some energy. The best thing to carry would be a fat or triglycerides because you'll get the most energy per pound. More energy than a pound of protein or more energy than a pound of carbohydrate. Any question about any of that? All right, let's briefly talk about phospholipid. This is the one example of a lipid which is a polar molecule which can dissolve in water. A phospholipid has a phosphate group attached to the third position of glycerol. So here we have glycerol right there. There's the phosphate group. And it does have two fatty acids attached to it. The third position by the phosphate group. That phosphate group, as well as the glycerol, makes the head portion of this molecule polar. The tail portion, or the lipid fatty acid portion, is nonpolar. That makes the phospholipid a biphasic molecule that can dissolve either in water or in a lipid. We don't have many molecules which are biphasic, but you actually are familiar with maybe two of them, and that is soap and detergents. And the reason why soap and detergents help us clean, like our cleaner clothes in the washing machine, is that the water can dissolve any water soluble or polar molecules from our clothes, but it's not going to dissolve the lipids. And the detergent will dissolve the lipids, helping us to clean our clothes. Any question about any of that? All right, the phospholipid is the main molecule in our cell membrane, and it just gets together like that, which then in water will form this, and that actually looks like a cell. Think of this as a red blood cell in the blood, which is mostly water. The heads, or the phosphate portion of the molecule, facing outside towards the water, and then the heads facing inside in the lipid membrane, facing the cytoplasm, which is mostly water. And then the phospholipid tails, meaning the fatty acid portions, come inside the lipid membrane because they like to be together and they're nonpolar. And you put phospholipids in water, they'll first form a bilayer like that, and then they'll actually form the liposome like that. Water on the inside, the heads on the inside facing the water, the fatty acids on the inside facing each other, and then the heads on the outside facing the water outside. Any question about any of that? Let me talk about, well, let me just briefly state in our cell membrane, because we don't have a cell wall to help protect the cell membrane, we have cholesterol in the cell membrane to give the membrane strength and support. So right there is the cholesterol, and you can find it in between the phospholipid of the cell membrane. And you only find cholesterol in animals, cells, or tissues. You do not find it in plant cells. All right, any question about anything? If not, I'm going to end it here, and I'll see you in the lab at 6:32. I'm going to give you 2 minutes since I went a little bit beyond here. All right, if there's no questions, I'm going to log out, and I'll see you in the lab if you're in the lab at 6:32.